And now I would like to welcome on stage Thomas Kledal, who will introduce next team InProTher.
Thank you very much. I have been a coach for InProTher in this process here. Where are they? I don't know where they are. There, Peter and Nikolai. But really it's been a very, very easy job because they are so self-driven with Peter's high-level science and Nikolai's business background. It's actually been quite an easy task, and I hope it works out as you will see. InProTher is really all about making cancers visible to the immune system, hence, going directly into the immuno-oncology hype that is really spot on right now. Peter and I goes a long way back. So I think we've known each other for basically 20 years, published our first paper together 18 years ago or something like that. And back then you were a young, rebellious scientist. Now you are experienced in successful technology transfer and going with IP all the way to actually clinical studies with big pharmaceutical companies. You have done that. And it all starts with, really fascinating science on targeting retrovirally encoded DNA elements, but now actually showing how they work in tumor models in mice. And building this fantastic company with a focused approach to, for instance, breast cancer and head and neck cancer. I just want to say one last thing, is that when I look at you now, you are still a young and rebellious scientist, and I hope you will stay that way no matter how successful InProTher is going to be. So please welcome Peter on stage.
Yes. Am I on? Yes. Thank you, Thomas. That was a good introduction. Yes, my name is Peter Holst. I have been doing vaccine research for the last 15 years, and my research and my team's research has entered into InProTher now, where we are trying to work with immunotherapy. So immunotherapy is a very exciting discipline within science, because we now know that it's possible to cure even late-stage cancers. Immunotherapy can currently cure a few cancers, a tiny bit of the total spectrum of cancer. But we can see rationales that maybe many more could be cured in the future. However, the majority lies out of reach of those treatment principles currently being advanced. So in order to get to those, we need to understand what kind of tricks are cancers using to hide from the immune system. Now, the trick that we have discovered in old literature has basically come from our ancestors. Monkeys, early humanoids, were infected by retroviruses. These retroviruses stayed in our genome, and now 8% of the genome of modern humans consist of ancient retroviral DNA. These elements are necessary for cancer development and always overexpressed in cancers, with a few exceptions of virus-induced cancers. Now, what's this connection then? So the immune system obviously is designed to kill viruses. So the viral infection of a host cell has in common the introduction of a viral genome into the host cell, and that introduction is a powerful immune stimulus. Any successful virus must have found a way to counteract this. And the trick that retroviruses use is actually to have a patch, a sequence within their attachment protein that binds to host cells and injects its nucleus or its nucleotides. That patch is inhibiting the immune response towards the viral genome. Now, we take this fusion protein and we mutate the immunosuppressive patch and insert this into a clinically proven, very potent immunostimulatory vaccine vector. And the result is a potent vaccine that can direct a powerful T cell and antibody, basically the cellular and the humoral immune response towards the cancers. Once we have done that, cancers are now targeted by their own immune evasion mechanism, and now they're vulnerable. Now, this is probably the only primary data I'm going to show, but what you see here is basically an example of a therapeutic trial against colorectal cancer, where the animals that are being vaccinated in the black are rapidly clearing the cancers, controls are growing. And what we did to prove the generality of this approach, I think this is good enough really, but to prove the generality, we took months later and challenged these mice not with colorectal cancer again, but with the breast cancer in the mammary glands. None of the animals could engraft a new breast cancer. So this is a curative and prophylactic tumor immunity. Now, the potential of such an approach is obviously enormous. more than 1 billion people today will die from cancer, at least unless we do something about it. Now, we're not there. We are actually at a stage where we're trying to get into human trials and prove our point in humans. But with a general approach, there are many options for us. We can see that other companies who have succeeded in generating early-stage clinical data have made either billion or multi-billion-dollar evaluations and exits. And this is what we are trying to get into. Now, we went out with this data package to the scientific community in Denmark, in Europe, in the US, and the response was uniform. They all loved this data package. And actually, all of them wanted to be part of it. Now I'm showing you a quote from Jay Posofsky, the head of the vaccine branch of the NCI, in the summary stating that, could I play a part in this? And this is the feedback we get. Now, with the support of the BAA and this kind of feedback and this kind of support from the community, we've been able to establish a good team to take this forward. Now, I am the only person actually in the world that has succeeded in making better immune responses to viral vectors as they are. I can increase the response of viral vector vaccination and have that track record. Nikolaj Høyer Nielsen is a serial entrepreneur and a guy who really can take companies up and get them moving. And he's been doing this, and he will continue to do this in the future. We have been able to recruit Anne-Marie Anderson from Oxford. She has a background in having developed this core platform technology that we're going to use, and it's a perfect profile for our platform development. Future employees, we can recruit a really experienced CEO who has field-specific business development experience for many, many years, hundreds of deals to our company. So we have the business platform in place. And we can also recruit the animal model specialist and the oncology trained staff we need to go forward. We have access to supports in biotech companies specialized in vector development. And for our later development, we have access to Marie Svane and Per Thor Straten at the Center for Cancer Immunotherapy, which of course said, can we run your trials? So that support is there as well. Now, if you look at our progression in the BAA, yeah, this is a stereotype, but I think it holds true. We had basically HIV and cancer and whatever on the palette a few months ago. And now we have a focused approach to target cancer and get this into humans. Our approach has been validated by Big Pharma. We got positive feedback from investors. We have freedom to operate. We own our own IPR. We can assemble the team. What we need now is the research infrastructure and the financial support. And then we can make rapid advance towards clinical trials and basically into the market. Thank you.
Thank you. Thank you, Peter. Do we have any comments from the panel?
Well, thank you, Peter. Very good presentation. Obviously, I don't know how many presentations you've given to me over the years, but it's certainly a lot. And I do appreciate that how the focus that has been brought into your project, as the mad scientists there indicate, there's always been a lot of ideas. So great, you are now concentrated on the immuno-oncology approach. My question then is, given that immuno-oncology is a very crowded space, a lot is happening there. There's more than 1,000 combination trials currently going on with all kinds of therapies. How do you see this approach fitting into the clinical practice?
I'm so glad you asked that because I was saving some time and didn't explain it myself. So basically, there's a fantastic hub now in targeting cancer-specific mutations. This is the majority of the hub. We don't do that. We target a shared fundamental principle of cancer development. And we're basically separating us from the vast majority of this hub. Now, they can only target basically in very few cancers that are not smoke and sunlight induced. They need mutations to do that effectively. And we are aiming much broader than that. I can also say that we have reproduced past failures. So I found the first immunotherapy trial using viral sequences back in '81. If we make this kind of approach without introducing our prime mutations, we cannot direct immune responses against these viral genes. They are immunosuppressive. So we have basically proven the difficulties, but also solved them. And I guess this difficulty in getting a response up may explain why cancers have stayed vulnerable to endogenous retroviruses, even in their evolution. I think we stand apart by targeting a different segment of cancers, by very, very good preclinical data that I think most would be envious about. And then this broad cancer approach that we basically can find our best position. If we get sufficient funding in, we have a platform technology now that targets functions that we can see are essential for cancer immunization. Unlike other checkpoint inhibitors right now, we're actually talking about cancer-specific immune checkpoints. So I think the differentiation is strong enough. What we need now is more support to build a stronger data package and develop the platform.
I appreciate this is sort of the first step of an answer, but I do also think that the world in oncology is super complicated, and that there are other projects doing similar or at least comparable things. It's not all of the projects. But all right. But what would you? What is your current funding requirement? What are the next sort of key milestones for this project? Where would you like to see sort of the next critical experiments go?
Right. I mean, we will be tremendously happy to enter the BI format here, because we have a few things to address. We would like to go in clinical trials with the ability to stratify patients based on our target selections as a one key thing. So right now we'll be happy to raise the next 10 million kroner from BI and maybe some extra funding. But very rapidly we'll be looking for the funding required for our clinical data package. And we have a hurdle. We're using viral vectors. They're expensive tools. And we need 3 million just for the GMP and the tox. So we're probably talking about 10 million euro after this phase, which will start hopefully after getting some money from the BI.
And then thank you also for me. Excellent presentation.
Thanks.
Very short. What is it that makes you sleepless during night? What is the major challenging you're facing right now? Is it finance, infrastructure, or is it on the science part?
Well, I have no funding next year. So I have basically stepped all the way into this. It is not true. I have 20% at KU next year, basically to have an easy access to animal facility. So I'm gambling all on this, and the deadlines are getting close. So that is an issue, of course. I think we're on a good track. I hope you all agree, and I hope that the BI agrees, and then we're fine, and I shouldn't lose any sleep. But from that, we're talking about a new class of immunotherapy, and I really want to see this work in humans. I mean, every clinician we talk to say this is data package deserves to be in human trials, and obviously until we are there, it may not work.
Thank you very much, Peter.